Ariel received his PhD in record time in chemistry at Yale University, working for quite a famous quantum chemist named Immuno. He then moved to the Max Planck Institute, where he worked with Manfred Eigen, and eventually made his way to Texas, where he's been for over the past decade. He is originally from Argentina, and his research has taken, while he's classically trained and is extremely knowledgeable about biophysics and statistical mechanics and simulation techniques, his recent research has taken a very interesting direction, and he's trying to understand, which is a key problem, the origin of drug specificity. Because obviously, most drugs fail not because they kill you. The pharmaceutical companies are now pretty good, at least on average, at not giving you something in phase 1 clinical trials that is going to make you die. But they don't work. There's a lot of side effects. And so the key question is what is the nature of these side effects? Can one understand it? And can one predict it a priori in the design process, so you just don't spend $1 billion on your favorite molecule only to discover that not only does it treat disease X, it kills you because it interferes with disease Y. Or worse, it turns things on and off, which could either be adventitious or not. And he's developed some very novel, extraordinarily insightful, and depending upon the community, controversial ideas, which makes it interesting. All new ideas tend to be met with resistance, which is good. Some will survive, some won't. And he's been extraordinarily creative, and he's very interested in the notion that he's going to be telling us about the nature of hydrogen bonding and a wrapping of structures. And its importance, hydrogen bonding, for those of you who don't know about proteins, is one of the most singular important interactions, dictating the physics of protein structures and probably the physics of interactions. It's both orientation and distance dependent, and it is big. It's one of the few interaction terms you could describe where you know that if you don't have a hydrogen bond, then you ought to. You ought to run the order of kcals per mole, which could be the whole binding free energy. And so today he's going to be talking to us about evolutionary insights into the control of drug specificity. It's a real pleasure to welcome Ariel. Thank you for coming to visit us.
Thank you very much for your kind hyperboles, Jeff. And it's very exciting to be here today with this very friendly and yet high-powered institution. So I'm going to, as the title indicates, I'm going to talk about the control of drug specificity, which, as you know, is a major problem in drug discovery and quite a nightmare in the endeavors of big pharmas. So from a rather oversimplified perspective, there are two core problems that one has to deal with addressing side effects. One is the fact that proteins evolve from each other, and hence the target is typically part of a family of proteins that share a common ancestor, and hence the structures are uncannily similar, right, of different members of the family. They're the so-called paralogs. So there's a great structural similarity, which makes it difficult to be selective when many things that you don't want to hit look very much alike your target. And the second major problem is that the target protein plays different roles in different cellular contexts. That means it may be okay to hit a particular target or to inhibit a particular biochemical function in one type of tissue or cell, but not in another. And of course, that may end up causing side effects, the context dependence of these target roles. So in regards to the structural aspects which relate to the first problem, here is an illustration of the type of problems that the community is facing when trying to develop effective drugs. So, for example, you have on that display aligned the ribbon structure of two proteins, two kinases, two important signal transducers of the cell. One the focal adhesion kinase, which is a major target in cancer metastasis, right? I mean, that's a transducer of adhesion signals. And the other is the insulin receptor kinase, which it's functionally completely unrelated to the former, but structurally very similar, in particular in the ATP binding site. And in fact, there's a high level of amino acid conservation in the region where the competitive inhibitor is going to be targeting, right? And so you can imagine that if you want to achieve specificity, you would be walking on thin ice because you want to hit the focal adhesion kinase, but you never want to touch the insulin receptor kinase because that would induce a diabetic coma in the patient, right? So that's absolutely, it would just severely compromise the glucose uptake of the erythrocytes. And so you don't really want to touch the insulin receptor. So how do you tell them apart if they are structurally so similar? And in fact, members of the same family within the superfamily. So one day I just decided to try to answer the question, what is it that distinguishes paralogs and we can take advantage of to make more specific drugs? And it occurs to me that the structure is not going to be the answer, and but maybe some structural attribute which can be derived from structure provides the clue. So I just decided that we probably needed to characterize the quality of the fold. So two proteins may have a perfectly alignable structure if they are close enough homologs, but the quality of the fold, as determined by the way the side chains protect the structure, may be quite different. Protect the structure from what? From from hydration of the backbone, which is, of course, the major destabilizing force of a native fold. So that takes me to re-examine hydrogen bonds, not so much as a pairwise interaction, but more in regards to the environment where they are formed. As you can imagine, a hydrogen bond formed, which is mostly electrostatics, it's 78 times stronger if it's formed in the vacuum than if it's formed in completely surrounded by water, right? Because, well, that's the difference in the permittivity between vacuum and water. And so if a hydrogen bond in the protein is going to prevail, then it better be quite dry in water, right? So although it may be a structural determinant of a soluble protein, it needs to be fairly dry if it's going to become a true structural determinant. So hydrogen, upon collapse of when the hydrophobic collapse takes place in a peptide chain, then you are also burying the backbone. So not any kind of collapse qualifies as such, because only a hydrophobic collapse that would actually lead to the formation of backbone-backbone hydrogen bonds is likely to prevail, because otherwise the cost of dehydrating amides and carbonyls would be unbearably high, compromising the stability of the compact structure. So the compact structure of a protein is achieved by a delicate compromise between the number of hydrogen bonds that the backbone is able to make with itself and the accretion of nonpolar groups that eventually can actually separate themselves properly from water through the so-called hydrophobic effect. But then you may ask, well, how well made are all these hydrogen bonds? What are their chances that they will survive? Is it possible that the cooperativity needed to protect the bond could fail sometimes in some bonds creating certain structural vulnerabilities. For example, if a bond is partially exposed to water, and then backbone hydration may actually lead to a partial dismantling of a structure, or local dismantling of a structure. So then, so one way of assessing the microenvironment of these bonds is simply to count the amount of nonpolar groups that surround them, right? So that would be much better than having to model the water or whether water can get in or out. So you could just simply count the number of nonpolar groups surrounding the bond within two spheres centered at the alpha carbons of the residues paired by the bond. And then if you take an adequate radius of those spheres, pretty much the cutoff distance for electrostatics in water, then you can actually draw some statistics and determine how many nonpolar groups from side chains are within those spheres, and then you have an idea of how dry or wet those hydrogen bonds are, in a way how vulnerable to water attack they are. And then some quite striking features arise from those statistics. So for one thing, the hydrogen bond is typically wrapped by 26 plus or minus 7 nonpolar groups. And then when you are in the tails of the distribution, in the lower tail of the distribution, you are dealing with an underwrapped hydrogen bond, with a bond that doesn't have enough, that's not dry enough in water. It's a vulnerable hydrogen bond. And it turns out that those are sticky and they distinguish paralogs. So they share two essential features that make them quite an interesting targetable feature for drug discovery, especially if you want to build specific drugs with controlled and high specificity. So the idea would be, the paradigm would be to try to design drugs that are actually contributing with nonpolar groups to enhance the stability and the electrostatics of a preformed hydrogen bond. This is a three-body effect, as you can imagine. So here is an example of these dehydrons, as we call them. Hydrogen bonds that are prone to get further dehydration by dragging on nearby hydrophobes in order to reduce their local dielectrics, or in order to enhance the preform electrostatics. So for example, here is a very curious feature of the p53 DNA binding domain. p53, as you know, is a crucial oncogene, right, because of its ability to police the integrity of the DNA and send the right, or create the right apoptotic signals in case the DNA is deficiently assembled. And so the region that interdigitates with the major groove of the DNA is actually a region deprived of tertiary structure. It's a helix that is sort of protruding from the structure like that, but it doesn't have tertiary structure. So you can immediately sense that there's something wrong with this region, right? In other words, if it doesn't have tertiary structure, that means it's probably not going to be able to wrap the bonds, the hydrogen bonds that make the helix, that are going to keep the helix in the place. So you know that there's a quote-unquote failure of cooperativity in this part of the structure. Or you may say there's a vulnerability there. And sure enough, this backbone hydrogen bond is actually quite vulnerable to water attack. But on the other hand, that water is not really a happy camper because it has lost some coordination, right? Instead of having coordination 4, as in bulk, it has now coordination 3. So that means this water molecule is probably quite easily expelled. It's there because there's room for it to stay there and hydrate the carbonyl, but it rather not be there, right? So these dehydrons are really a region of tension, you could say, of even interfacial tension, in that water can get there, but at a cost. right, at an entropic cost. In other words, it's lost its full coordination. And at the same time, the hydrogen bond is not really happy there. So given the chance that this hydration process progresses, then the hydrogen bonds will be dismantled and the amide and carbonyl will just simply be fully hydrated. On the other hand, if the right ligand comes in, then that would actually maintain the structure, because then you can actually, if you exclude the water, you can stabilize the bond and also enhance its electrostatic interaction. So in essence, we are looking at drug ligands as dielectric modulators here. That's sort of the paradigm we are after, and we believe they obey that these underwrapped hydrogen bonds are actually creating a dehydronic field, which is the mechanical equivalent of the propensity to get dehydrated, because as a hydrophobe approaches, it lowers, you would say in broad terms, in rather vague terms, it lowers the dielectric or the permittivity, and that would actually enhance the electrostatics. So this is not a conventional way of designing drugs, because you see, nobody would think of approaching a hydrophobe to a polar, but if a polar is interacting with another polar, then the hydrophobe has a business approaching it, because it can enhance the pre-existing Coulombic field, the Coulombic interaction. So these three body effects are a little bit similar to what happens in human relations. Sometimes you don't like somebody, but if a third person likes her, then you start liking her too, right? So it's a bit triangular. Okay, and so let me just now describe to you how these vulnerabilities of the protein, these dehydrons, behave in evolutionary terms. So the dehydron patterns behave in evolutionary terms. So if you look at, say, a particular protein or function, for example, the dehydrofolate reductase, let's say, a typical enzyme, widely studied, metabolic enzyme, widely studied, you look at it across species that have diverged billions of years ago, and you notice something quite striking, that a pattern that is always prevalent, which is the larger the population of a species, the better made the protein is. So the topology is pretty much the same, right, across species. But the wrapping is different. The wrapping is getting worse, right? So, sorry to say, but our proteins, regarded as subunits, are lousier than those of bacteria, and which is in turn lousier than those of archaea. Why? Well, essentially because selection is far more inefficient in human than it is in bacteria or in archaea, right? We have a much smaller population, and hence our probability of fixing mildly deleterious mutations in the population is much higher in human than it is in species with 10 orders of magnitude larger population, right? And so as a result, our proteins are more loose. But that could be also a blessing in disguise, because that means that this protein is far more reliant on binding partnerships to maintain its structural integrity, and in fact it fosters those complexations. It promotes those complexations because, as I told you, these packing defects, the dehydrons, are actually sticky. And hence, because they promote their own dehydration, they would tend to open up niches for the development of protein-protein interfaces, right? So the fact that this protein is lousier than this, or more loose in a way than this one, opens up the possibility of higher level of complexity. So the origin of that complexity is actually non-adaptive at all. It just stems from the simple fact that selection is far more inefficient in higher eukaryotes than it is in prokaryotes or in archaea. So the same happens now. So I examine what happens across orthologs, right? The same protein in different species. Now, what's of direct interest to the pharmacological industry is what happens across paralogs, right? So homologs within the same species. That's where the possibility. That's where we have to try to tell them apart, because if cross reactivities arise, that may lead to side effects, which is precisely what we are trying to avoid. So in paralogs, the story is a little different. So the paralog arose from a gene duplication event, right? So a gene duplication event produced a dosage imbalance, because let's say the gene, the product operates in a complexation, right? And now I have twice as much population or concentration of the gene product after the gene duplication event, and that is not properly titrated stoichiometrically with the concentration of the binding partner, which remains at the same concentration as before the gene duplication event took place. So that creates a dosage imbalance. Now, if a protein is highly reliant on binding partnerships to maintain its structural integrity, if it's not a loner, well, then that's going to have fitness consequences, because now you have a protein that needs the partner but doesn't get it, right? Doesn't get it in the right amounts. And so, and that's going to be quantified by the wrapping quality. The poorer the wrapping, the more reliant it is on binding partnerships, and the higher the fitness consequences. And so there are two solutions to that problem. So either one of the copies of the gene is simply selected out if selection is sufficiently efficient, or sub-functionalization will take place. In other words, there's going to be some level of paralog divergence due to the accumulation of mildly deleterious mutations that's going to change the wrapping of one copy with respect to another, or the expression pattern, or the post-transcriptional regulatory patterns that are recognized by the microRNA. But some level of divergence has to occur to mitigate the dosage imbalance, right? And we can harness on that divergence to create better drugs, because we know that in part that divergence is going to translate into differences in wrapping across paralogs, right? I mean, that's a fine-tuning of different functionalities for proteins of common ancestry. And because we know that wrapping defects are sticky, and that paralogs have different patterns of wrapping defects, we can create better drugs, right? More specific drugs that can actually tell apart different members of a family. Likewise, because paralogs will tend to express themselves differently, distinguish their expression pattern across tissues, another way of mitigating the dosage imbalance, then that opens up another opportunity for the drug designer to create tissue-specific delivery. And so, for example, if you want to tell apart two paralogs, one you want to touch and the other you don't want to touch, and the drug cannot avoid to have that, it has to be cross-reactive, because, well, your drug happens to be cross-reactive. You can still manage to distinguish them by tissue-specific delivery, right? So you know that one paralog, the one you want to touch, is expressed here but not there, and so you just direct your cross-reactive drug to that particular tissue, and then you ensure that you're going to be able to tell apart the paralogs. So, in essence, the paralog divergence arising essentially from the dosage imbalance and its fitness consequences can open up opportunities for better drug design based on this evolutionary observation. So, for example, here is how it works. Remember, I showed you the two examples early on of the focal adhesion kinase and the insulin receptor. structure, very similar. The root mean square displacement less than an angstrom for the backbone atoms, but a different pattern of dehydrons at the active site. So perhaps one could take advantage of this difference to create a drug that can actually tell apart the focal adhesion kinase and the insulin receptor, and that would be of great interest to the pharmaceutical industry because it would enable you to treat metastasis without the risk of inducing a diabetic coma on the patient, So this is the paradigm, although that's, you have to excuse my pomposity here. I shouldn't call it really paradigm. This is my suggestion. Let's call it like that for now. So the way people would go about expanding a lead typically it relates to what kind of targetable groups there are on the surface of the protein that they can match, right? So they say, oh, there's a exposed non-polar group on the surface. So, okay, so we'll try to put a non-polar on the drug, right, to match it and to enhance affinity. And that's fine. Nothing wrong with that, except that these features are typically highly conserved across paralogs. And so if you try to target them, or likewise, if there's a proton acceptor, you put a proton donor, or proton donor, put a proton acceptor. That's conventional thinking. What's wrong with that is that you are targeting features that are highly conserved, and hence you are promoting promiscuity. So that's you see, selectivity and affinity are two completely different ball games, and you don't want to target the low-hanging fruits are the forbidden fruits here, in other words, because these are the obvious things that a pedestrian designer would do, but they would lead to promiscuity. And so what I suggest to do is just try to introduce non-polar groups that would actually improve the wrapping of pre-existing hydrogen bonds, the dehydrons, which are not conserved across paralogs. And that would be a feature promoting specificity. So this is how it works. So for example, let me just give you a test example. So here are the crucial structural parts around the ATP pocket of the focal adhesion kinase and the insulin receptor. And you see, in green, you see an under-wrapped hydrogen bond, a dehydron, and gray denotes a well-wrapped one. So there's a wrapping difference here that distinguishes those two proteins. So we took an inhibitor of the focal adhesion kinase produced by Novartis called TAE226. So this inhibitor is never going to see the light of day because it induces diabetic coma in the patient. So you're never going to see it with the approval. But we just modify it to increase the level of the wrapping capability for this particular dehydron in the focal adhesion kinase, which, as I told you, aligns with a well-wrapped hydrogen bond in the insulin receptor. So that amounts to just do some synthetic chemistry, starting with a different reactant, and you end up having the methyl where you want it to be. And here is the assay for the insulin receptor, the immunochemistry for the insulin receptor kinase, which, as you can see, the TAE compound knocks out phosphorylation of the insulin receptor, but our compound, which just differs on the previous one in only one methyl, doesn't touch the insulin receptor. You see? We don't touch it. And these cells are glioblastoma cells, the kind of cancer that ended up killing Ted Kennedy, which are, of course, highly expressive of the insulin receptor, right? The brain uses a lot of glucose to function. And so this, and glioblastoma uses glucose because it has metabolism in overdrive. It uses even more so. So it has a lot more insulin receptor. And so these two type of glioblastoma cell, you see, TAE knocks it off and knocks it out, and our compound doesn't touch it.